Welcome to G Fun Facts Online. Today, we are stripping away the modern world. Forget the blue sky, forget the green forests, forget the birds and the mammals. We are going back 2.5 billion years to stand on the surface of an alien planet. It is Earth, but it is unrecognizable. The sky above you is a hazy, thick orange, choked with methane and carbon dioxide. The sun is faint, significantly dimmer than it is today. And the oceans? They aren't blue, they are green, heavy with dissolved iron, and the land is a jagged, rocky wasteland, utterly silent, whipped by fierce winds because there is no vegetation to slow them down. Life exists here, but it is a silent empire. It is a world ruled entirely by the microscopic, single-celled bacteria and archaea, living in an anaerobic world, a world without oxygen. But deep in the shallow, sunlit waters, a biological machine is coming online that will trigger the greatest catastrophe and the greatest miracle in planetary history. Today we are doing a deep dive into the Great Oxidation Event and the origin of you. Let's begin. To understand why you are here, listening to this, we have to understand the most dangerous waste product in the history of biology, oxygen. Today, we think of oxygen as the elixir of life. We panic without it. But for the first half of Earth's history, oxygen was a deadly poison. The dominant life forms on early Earth were anaerobes. To them, oxygen was a corrosive, reactive chemical that shredded their enzymes and destroyed their DNA. The atmosphere was chemically reducing, rich in methane, and essentially devoid of free oxygen. Then, roughly 2.5 billion years ago, a group of microbes called cyanobacteria stumbled upon a metabolic magic trick. For millions of years, life had relied on simple chemical sources for energy. But cyanobacteria figured out how to tap into the ultimate power source, the sun. They developed oxygenic photosynthesis. They learned to split water molecules to harvest electrons, powered by sunlight. It was an incredible innovation, but it had a catch. The process released a byproduct, oxygen. At first, this didn't matter much. For hundreds of millions of years, these cyanobacteria puffed out little whiffs of oxygen into the ocean. But the Earth acted like a giant chemical sponge. The early oceans were filled with dissolved ferrous iron. As soon as the oxygen was produced, it reacted with the iron. It's simple chemistry. The ocean rusted. This rust, iron oxide, was insoluble. It rained down from the water column, settling on the seafloor in massive, striped layers of red and black rock. These are the banded iron formations, or BIFs, that we mine today. If your car is made of steel, that steel likely came from the rusted oceans of 2.5 billion years ago. But eventually the sponge filled up. The iron was exhausted. The chemical sinks were saturated. With nowhere else to go, oxygen began to bubble out of the oceans and into the atmosphere. This is the moment the world ended. We call it the Great Oxidation Event, or the GOE. For the native life of Earth, this was an apocalypse. As oxygen levels rose, it triggered a mass extinction of the anaerobic microbes that had ruled the planet for eons. They had no defense against this reactive gas. It burned them at a cellular level. The survivors were forced into the shadows, retreating into deep ocean muds, hydrothermal vents, and oxygen-depleted pockets where their descendants still hide today. But the biological devastation was only half the story. The geological consequences were even more terrifying. At this time, the Earth was kept warm by a blanket of methane, a greenhouse gas 30 times more potent than carbon dioxide. When the rising oxygen met the methane in the atmosphere, it reacted, scrubbing the methane out and converting it into carbon dioxide and water. Carbon dioxide is a greenhouse gas, yes, but it is much weaker than methane. The result was a catastrophic collapse in global temperatures. The Earth plunged into the Huronian glaciation. It's likely the planet became a snowball Earth, with ice sheets stretching from the poles all the way to the tropics. Life hung by a thread, trapped under kilometers of ice on a frozen, poisoned world. However, life is resilient. The Earth eventually thawed, and the oxygen levels stabilized. We entered a period that geologists, somewhat unfairly, call the Boring Billion. This era, lasting from about 1.8 to 0.8 billion years ago, was geologically quiet. The oceans were stratified, with oxygen on the surface and toxic, sulfur-rich, oxygen-free waters in the depths, a condition known as a Canfield Ocean. But biologically, the Boring Billion was anything but boring. It was the crucible where the second part of our story takes place. This is where life stopped just existing and started innovating. This is where eukaryogenesis happened. Let's pause on that word, eukaryogenesis. It sounds technical, but it is the origin story of every plant, animal, fungus, and human. Life on Earth is generally divided into huge domains. For a long time, textbooks taught the three domains model: bacteria, archaea, and eukaryotes. Bacteria, you know. Archaea are single-celled organisms that look like bacteria, but are chemically very different, often living in extreme environments. And eukaryotes are us, organisms made of complex cells with a nucleus and organelles. The mystery was always, where did eukaryotes come from? We are so complex compared to bacteria. A bacterial cell is like a studio apartment. A eukaryotic cell is like a sprawling mansion with specialized rooms, a library, and a power plant. Did we evolve separately? Did we branch off from bacteria? In 2015, the textbook was rewritten. A team of researchers led by Tejas Etema was analyzing mud samples from a hydrothermal vent field in the Arctic Ocean, a place called Loki's Castle. In that mud, they found DNA. It belonged to an archaeon, but it was weird. It had genes that were supposed to be unique to us, to eukaryotes. Genes for building internal skeletons, for bending membranes, for trafficking cellular garbage. They named this organism Loki archaeota. Soon, they found more relatives. Thor archaeota, Odin archaeota, Heimdall archaeota. Collectively, they are the Asgard archaea. And the genetic analysis revealed a stunning truth. Eukaryotes did not branch off from archaea. We emerged from within them. The family tree isn't 3 branches. It's 2. We are, at our fundamental core, complex archaea. You are a direct descendant of the Asgard superphylum. But simply finding the ancestor doesn't explain the mechanism. How does a simple Asgard archaeon living in the mud turn into a complex human cell? This brings us to the most elegant theory in evolutionary biology, the hydrogen hypothesis. We know that almost all eukaryotic cells have mitochondria, the power plants that generate energy. We also know, thanks to the work of Lynn Margulis in the 20th century, that mitochondria were once free-living bacteria. They have their own DNA. They look like bacteria. But why did the host cell merge with the bacterium? The old story was that the host tried to eat the bacterium and failed to digest it. But that's a bit too random. The hydrogen hypothesis, proposed by William Martin and Miklos Müller, offers a reason based on metabolic necessity, a trade deal. Picture the boring billion. You have 2 microbes living close together in the mud. First, you have the host, an Asgard archaeon, likely similar to Heimdall archaeota. It is an autotroph, meaning it builds its own food, and it relies on hydrogen gas for energy. Second, you have a bacterium, specifically an alpha proteobacterium. This bacterium is versatile. It can breathe oxygen if it's around, but in the low oxygen world of the deep ocean, it often switches to fermentation. And what is the waste product of fermentation? Hydrogen. See the connection? The archaeal host gets addicted to the hydrogen waste of the bacterium. It's a syntrophic relationship. One feeds the other. To maximize the harvest of hydrogen, the archaeal host starts to wrap itself around the bacterium, increasing the surface area of contact. Over generations, this physical embrace tightens. The host doesn't just touch the bacterium, it engulfs it. It pulls the bacterium inside its own body, not to digest it, but to farm it. It effectively creates a hydrogen generator inside its own living room. This was the moment of eukaryogenesis, the moment life changed forever. Once the bacterium was inside, the relationship shifted. The world was slowly becoming more oxygenated. The internal bacterium was capable of aerobic respiration, using oxygen to burn food. This is vastly more efficient than the old anaerobic methods. Anaerobic fermentation gives you 2 units of energy per molecule of glucose. Aerobic respiration gives you 36. It is an 18-fold increase in power. Over time, the bacterium lost its independence. It transferred most of its genes to the host's control center, which became the nucleus. The bacterium became the mitochondrion. And this energy surplus is the key to everything. Bacteria are limited by a geometric constraint. They generate energy across their outer membrane. If they get too big, their volume grows faster than their surface area, and they can't make enough energy to support their size. They hit a glass ceiling. They cannot become complex. They cannot build large genomes or complex machinery because they simply can't afford the energy bill. But the new eukaryotic cell? It had broken the glass ceiling. By internalizing the power plants, it could add as many mitochondria as it needed. It had a fleet of internal generators. Suddenly, the cell was awash in energy. It could afford to expand its genome by orders of magnitude. It could accumulate DNA that wasn't strictly necessary for survival, but could be used for regulation and complexity, the so-called junk DNA that actually runs the show. It could build dynamic internal skeletons using the actin proteins inherited from its Asgard ancestor. It could transport cargo across vast distances inside the cell. This merger allowed life to escape the microscopic. It allowed for the evolution of multicellularity. It allowed for specialized cells, neurons, muscle fibers, blood cells. None of this is possible without the massive energy surplus provided by the mitochondrion. So when we look back at the Great Oxidation Event, we see a paradox. The oxygen produced by cyanobacteria was a poison that wiped out the ancient world. It caused a global freeze, but that same oxygen, once the dust settled, became the high-octane fuel for the new chimeric life forms. The Asgard host provided the complex genetic toolkit, the skeleton, the membrane manipulation. The enslaved bacterium provided the power source. Together, they conquered the planet. It is a humbling realization. We tend to think of ourselves as a single entity. I am a human. But you are not one thing. You are a colony. You are a Russian nesting doll of biology. Every cell in your body is a scene from an ancient merger. Your DNA is mostly archaeal. Your energy is bacterial. You are a chimera stitched together 2 billion years ago in the dark, sulfurous mud of a boring billion ocean. Every breath you take right now is a tribute to that history. You breathe in the corrosive waste product of cyanobacteria, oxygen. Your lungs send it into your blood, which carries it to your cells. Inside your cells, the descendants of ancient bacteria, your mitochondria, take that oxygen and burn it to keep you alive, powering the electrical signals in your brain that allow you to understand this very sentence. Life didn't just adapt to the Earth. Life remade the Earth, destroyed it, and then built a new world from the ashes. We are the children of that reconstruction. From the poisoned sky to the frozen Earth, to the merger in the deep, our history is written in the very chemistry of our cells. And that is the fun fact for today. Thanks for listening to G Fun Facts Online.